Good evening for everyone. Now we start the last presentation for the International Symposium in Veterinary Science, and I invited Dr. Guilherme Klafke, PhD, Research at Animal Health Research Center, and he has expertise in the following areas: animal health, vectors and vector-borne disease, resistance to antiparasitic drugs. Thank you very much for your participation. And now you can share your screen and start your presentation.
All right. Thank you, Viviane. Thank you for the invitation. Thank you for the organizing committee to open the opportunity for me to present my work here. It's a pleasure to be here. It was a great afternoon with the talks. I believe Dr. Daniel Sonnenschein made a good preamble for my presentation. Actually, it's an honor to talk after him, not opening for him. But so I'm going to talk today about bioassays and in the development of molecular tests for pesticide resistance diagnosis in ticks. As Viviane said, I'm a veterinarian. I work at the Institute of Veterinary Research, which is part of the Department of Agricultural Research of the Secretary of Agriculture of Rio Grande do Sul State. So the IPV, this is the way we call it, it's a official laboratory for diagnostic of diseases of production animals in the state. We have a service for diagnosis for producers. We also work with OIE diseases diagnostics. So we have this research lines. Also, we have research work, and we do have service and extension services for producers in the state. I work at the parasitology lab, and the main work of the parasitology lab is based on tick research. So this is a 40-year work with ticks in the lab. So this is our main aspect. So I'm going to talk about ticks. Well, I believe Dr. Sonnenschein gave a better introduction about it. So ticks are arthropods. They're related to spiders and other arachnidae. They are acri, but they are all hematophagous. And we have three main families of ticks: the Ixotidae, which are the hard ticks; the Argosidae, which are Nutalilidae, which are some kind of how we call it, like a lost gap between the two big families. So it's a very diverse animal, and it can like affect a large number of hosts, mammals, birds, reptiles, amphibians. But the focus today will be in three main ticks. They are more related to the veterinary problem and also with human diseases. And those are first the most economic impactant tick for cattle producing in the world is the Rhipcephalus microplus, which is part of a complex of the species. It has three different species: Rhipcephalus annulatus, Rhipcephalus australis, and Rhipcephalus microplus. It's a one-host tick. It's a major vector of babesia and Anaplasma marginale to cattle. It has a huge economic impact. The other tick we're going to talk today about it's Amblyomma cagenesis species group. And a few years ago, there was a establishment of this species group. But we're going to talk today about these ticks that are more related to the cattle producing and human diseases, which is Amblyomma cagenesis, Amblyomma mixtum, and Amblyomma sculptum, being Amblyomma cagenesis and Amblyomma sculptum present in Brazil, and Amblyomma mixtum, more related to Central America, Colombia, and southern United States. And also, I'm going to talk a little bit about Rhipicephalus sanguineus complex. There's a lot of discussion about the phylogenetics and the speciation of this tick, but as far as the concern of this presentation, I'm going to call the brown dog tick or Rhipicephalus sanguineus tick without discussing the different aspects of its biology. But it has worldwide distribution. It's the vector of a number of diseases to dogs and to humans: rickettsia, babesia, anaplasma, ehrlichia, mycoplasma, and hepatozoon. It's a three-host tick. It can affect. It's not the three-host tick that requires different species, but it has this biology of 3 different hosts complete the cycle, and is present in most part of the world. So those 3 ticks are very important in terms of veterinary and human diseases. And we have in the market a lot of different synthetic acaricides to control them. Most of it are, at least fluazuron, is more used to, and organophosphate to control the cattle ticks. But the other drugs are available to use against the brown dog tick, and for amblyomma, of course, amitraz not in horses, but they can be used pyrethroids and microcyclactones. So we have several tools to control these ticks. And what happened is those different drugs, and they have different applications. They have a number of different formulations and modes of applications. We have puron, we have emulsifiable concentrates, we have injectable, we have so different ways of applying that. And what happened is that those most of these drugs, they act on the nervous system of the arthropods, right, of the ticks. So just in general, the synganglion is a major organ of the nervous system of the ticks. And we have sensorial neurons, and we have motor neurons, and we have muscles, so all these interconnections. And these drugs, they act on disrupting this nervous system, the good functioning of these nervous systems in some way. So the pyrethroids, they act on the neurons, on the axons by disrupting the sodium channels. The organophosphates, they act by blocking the acetylcholinesterases. The fipronil acts on the GABA chloride channels, amitraz on the beta-adrenergic and octopamine receptors, macrocyclactones on glutamate-gated chloride channels. So they all cause either hyperexcitation or depression, depending on the drug, depending on what they do on the neurons, leading to death of the parasite. So, this is fluazuron has a different mode of action. It disrupts the chitin synthase and interrupts the molt of the ticks from larvae to nymphs and nymphs to adults, and they disrupt the hatch of larvae from the eggs. But in the end, it caused the death of the ticks. So now leading to resistance, right? Well, in 1859, this guy named Charles Darwin, he published this book. It's called The Origin of Species. I believe most of you have heard about it. And there's an interesting thought in this book. And at some point he writes, can it then be thought improbable that other variations useful in some ways to each being and the great and complex battle of life should sometimes occur in the course of thousands of generations? We know that ticks can have several generations per year, so can be applied to ticks. So if such do occur, can we doubt? Remember that many more individuals are born that can possibly survive. This is the case of the ticks. Each female can produce thousands of eggs and thousands of larvae. It's a highly producive biological impact or biological potency, those ticks. that individuals having any advantage, however slight, of others will have the best chance of surviving and of procreating their kind. This preservation of favorable variations and the rejection of injurious variations are called natural selection. So the point is, can those ticks develop something that may lead them to survive in this struggle of life? So that's resistance. Resistance is evolution. So Dobzhansky is was a geneticist that wrote this book, Genetics and the Origin of Species, that he returns to the book of the origin of species and adds the Mendelian genetics on it and try to explain the Darwin theory by adding the genetics background on it. And he said that pesticide resistance is probably the best proof that of effectiveness of natural selection yet obtained. So it occurs in a different number of different species of pests, agricultural, medical, veterinarian, to a number of different pesticides. So it's there. It's evolution, and how it occurs in the field. So when you take an original population of ticks, and you never use a given pesticide, you have a few individuals that have the difference that allow them to survive to the treatment. So every time that you treat those populations, you eliminate the susceptible part of it and you favor the resistant ticks. And by eliminating it along the time, in the end, you have a resistant population that reflects in the efficacy failure. So basically, for acaricide resistance in the Americas, talking about Rhipicephalus microplus, which is the cattle tick, we have resistance to all 6 major classes of acaricides available. In Brazil, we have already registered resistance to all the classes. And we have some information that Uruguay also has resistance to fluazolam, but Argentina has the same problem, Colombia has the same problem, Mexico has the same problem. Some other countries are less affected, and we have a little bit of resistance in southern Texas, where we have ticks outbreaks there. But in general, we have a problem in South America that is very big. We also, regarding Rhipicephalus sanguineus, so the brown tick, we have reports of resistance to pyrethroids, to organophosphates, amitraz, and microcyclactones and fipronil. So we have this in Brazil, we have this in recently detected in the United States and Mexico. So it's part of the game, right? And for Emblema cagenaceae, the number of reports are much less. We have some reports in Mexico, in the coast, like the east coast of Mexico, and we also have a paper published with population in Goiás state regarding synthetic pyrethroid resistance and amitraz resistance in the species. So acaricide resistance diagnostic, why it's so important, right? So basically it's to confirm if some treatment failure that is being observed in the field is due to resistance, and to help on the decision in changing or rotating acaricides, and also to monitor for early development of resistance to a new product. So this is the major function or the major objective when we think about acaricide resistance diagnostics. Across along the history, we have, since the 1960s, we have people in Australia trying to develop methods for detecting those rare individuals that are resistant to the pesticides, and we have those classic papers describing the techniques that we use until now to detect resistance, right? And we passed later to the adult immersion test that was developed in the United States, and modification of those methods to detect resistance. So the most used one across the globe is an adaptation of Drummond's test, which is the adult immersion test. Basically, you collect ticks in the field, you have to collect a large number of ticks depending on the number of products you want to test, because you have to use 10 to 30 ticks per product. You use usually the concentration of the pesticide you use is the one that is written in the label, so you can test if that specific concentration is able to control those ticks, or you can use discriminating doses that are established at the laboratory, so we can separate populations that are resistant from the ones that are susceptible using technical products. They are more used to systemic or topical acaricides like microcyclactones, fipronil, and fluazurone. Depending on the product, you use different emission times, but in the end, you have 7 days for screening to see if those products work. But the final results obtained about 45 days after the collection of ticks, and you can do the analysis of the hatch, of the reproductive performance of those ticks, and how much eggs they produce, and if those eggs are viable or not. This takes time, right? About 45 days, and we make a calculation, and we establish based on the percentage control that is calculated based on index of fertility. You say if that population is susceptible. At our laboratory, we consider susceptible to a population to a given pesticide if the control is over 95%. Anything below that is suspected of resistance. At the lab, we receive about 100 samples of ticks from different locations in the state of Rio Grande do Sul, mostly from the western bank or western border of Rio Grande do Sul, which is the most important cattle producing area in the state, and receive a lot of ticks of that area from the central part of the state. And if we take the results from 2015 to 2019, we have that 98% of these populations are already resistant to cypermethrin, 80% resistant to amitraz, 30% resistant to organophosphates, and already 40% resistant to the mixtures of organophosphates and pyrethroids. So although immersion test is very important to give this a rapid, prompt response to the producers to say, hey, which product we're going to use in the next treatment. So they send the ticks to us, we test it, we screen it for resistance, and we use that as a method to give the answer back to the producer of which acaricide they're going to use. The other bioassays available are the larval tests. The larval tests are a little different. We can use immersion tests, or we can use packet tests. Basically, you impregnate a piece of paper with a filter paper with different concentrations of a given pesticide, and you do those response assays. You have to use a different number of concentrations there. So you can establish discriminating dose, establish LC50s, and compare populations. And again, to run this test, you need at least 45 days so we can have the larvae ready to go to do the testing. So the ticks, they have to lay the eggs have to be incubated, the larvae needs to hatch, and then we can run the test. So it takes time. So basically, these results, there's two ways of running this test. One's the dose response, which is quantitative test. When you compare the dose response of a susceptible population, a reference strain, and you have a population that you suspect of resistance, and you run tests, you do several concentrations, and you compare the dose responses. Right? This is a resistant strain. It's called a Jaguar strain, which is resistant to ivermectin. This is ivermectin test, larval immersion test, and you compare that with a Porto Alegre strain, and you can see the difference on the number of ticks that die that can survive at the given concentrations higher in a resistant population. And of course, you can do the LC50 calculations. There are several statistical softwares that can do this for you. And basically, you compare and you calculate the resistance ratios to see if a given population is resistant or not. And interesting, last week we have published this paper on the journal Insect Science. It gives a lot of good information about how to run a bioassay and how how to interpret results, and also give the codes for that R software, so you can program and run a probit analysis on those data. So it's a very cool and handy paper that came out last week. So just take a break for the mate, because so the other way to run a bioassay with larvae is to run discriminating doses. Basically, discriminating dose is that dose that we use that we can discriminate, the name says, we can discriminate between resistant population from 99% of that given population, and use that and double that to and run bioassays to see if a population is resistant or not, right? So here we have two different populations. One, we tested a battery of drugs here, and those are, you can see that this one is susceptible. So green is live ticks and mortality here in red. So it's completely susceptible to fipronil and ivermectin. It's a little bit of resistant to amitraz, it's susceptible to cunofos, and it's resistant to permethrin. And this is trait, right? It was resistant to all the 5 classes that we tested. So we ran this assays in Rio Grande do Sul, and we were able to show that we have overspread resistance to cypermethrin, amitraz, and more or less resistant to chlorpyrifos, ivermectin, fipronil. So it's a good tool for you to survey an area or a region or some farms and say what is the distribution of resistance on that specific area. So it gives good information in terms of monitoring and in establishing policies for control and decision making in terms of which drug we're going to use in different areas. So this is what the bioassays are for. Those tests can be applied to all the species of ticks, and this is how the people that all these papers here are produced by people that work with the cattle ticks, and they started to adapt those same techniques to other species like the brown dog tick. So in the past, like in the 2000s, we have the first work doing the characterization of sanguineous to acaricide resistance in Panama. Then we have the first description of resistance in Pseudomonas sanguineus in the United States to permethrin and fipronil. Then we have 2 other works in Mexico, and we published last year describing resistance. There's a paper from Goiás as well, and we have described the resistance in Rio Grande do Sul. So those techniques can be adapted to all the species, and the same has been done with the Myeloma caginense in Brazil and also in Mexico. So we can use the same ideas, we can do dose response, we can compare populations, we can do the same approach to detect resistance in larvae or adults in these 2 species. And we can also adapt, like in the case of those 2 species of ticks that they have more hosts, they're not one host ticks. We are also able to test nymphs or engorged nymphs or larvae, so we can manage that. So there's a lot of we can use the same techniques on both on other species. And this is the idea. With that, we can detect resistance, and we can drive the decision making of which pesticide we're going to use to treat the dogs or to treat the horses or treat an area in the case of the Myeloma caginense. So start change a little bit for the mechanism of resistance and the molecular markers. Well, why it's important to know the molecular the mechanism of resistance. So basically, all these mechanisms of resistance, they are molecularly detectable. So they're there in the DNA of the ticks, right? because it's an inherited phenomenon, so it's there in the DNA. When you think about bioassays, it can take 6 weeks of work. The molecular detection has the potential to give you results, and I'm going to give you a practical standpoint of that in about 1 or 2 days. But that doesn't mean it's not full. I mean, it's not. You need a lot of work to do that. You have to extract DNA, you have to standardize a PCR, you have to run a PCR, you have to run a gel, and you have to analyze the results. So there's a lot of work in both sides for us. In terms of resistant mechanisms of ticks, there's a lot of mechanisms that are already described in ticks, both target site alterations, which is when the target site of where the drug binds inside the nervous system, it has a difference, and the drug doesn't bind anymore, so it doesn't. The drug doesn't work anymore. Or you have the toxification. It's when the ticks, they have an enhanced membrane transporters that will pump out the drug out of the system. Well, on synthetic pyrethroids and a sodium channel model, which is the one that we know best. So what differentiates a susceptible and a resistant strain, right, is the genotype that leads to phenotypes. This is the central dogma of molecular biology. You have a genotype, and then you have a phenotype. And the way to get there, there's a different description, there's processing, there's translation, there's folding of proteins, and all that impacts on the phenotype. And we can use all this information that is on the processing of the genotype and phenotype, so we can detect resistance. We can do PCR, RT-PCR, we can sequencing those genes, or we can do sequencing, we can do transcriptome, we can do quantitative PCR to understand the expression of those enzymes, or the genes that code for those enzymes, or we can do analysis of those enzymes doing enzymatic activity, or we can do proteomics, or in the end we can do toxicological and functional tests to understand why and how those ticks are resistant or susceptible. So we have technology to understand that. Regarding pyrethroids, we know that there are several mutations that were already linked to pyrethroid resistance, and there's since the 2000s we have methods or technology to detect those mutations by PCR. And last year we published a paper about high resolution melting analysis for detection of these associated with resistance, which is a new way of detecting this resistance. I'm going to talk about it later. So, talking about the detection of mutations by using molecular diagnostics, I'm going to, just as a practical standpoint, I'm going to talk about a case that happened in Puerto Rico. There's a surveillance for acaricide resistance in Puerto Rico. There's a program for eradication there. Puerto Rico is an island that is a territory from the United States, and it's in the Caribbean. It's close to Cuba and República Dominicana. Beautiful place. So here in this area, it's called Yabucoa. There's a dairy producing farm there, and you can see the cattle being managed here. So on the 7th of November, ticks were collected on this specific farm. They have no idea of resistance there. They were using the BA86 based vaccine to try to control the ticks using macrocyclactones. Doing a management to eradicate the ticks on that property. And they collect the ticks and they send to the lab. They FedEx to the lab the next day, send it to Texas. We received these ticks like 3 days later, in a Monday. We received about 40 ticks. We separated 20 fully engorged females that were incubated to run bioassays. And I took this 19 semi-engorged females, so ticks that were not useful for bioassays because they were too small, they were too less engorged. So very tiny ticks that we can't use those ticks for nothing, right, in terms of bioassay. So I went to the lab, extracted the DNA, and then run the HRM, which is this PCR, which was like about 7 days after the collection. So here you can see in this table the genotype of each of those ticks, right? So, and how they compare with controls that we have. So we have a control for each kind of mutation, and with these graphs, this melting analysis of these fragments, we can compare and see which is the most likely genotype based on percent confidence. So after 7 days after the collection, we already knew that those ticks they had mutations associated with resistance to pyrethroids. So basically, 49 days after, so only in December, we got the results from the bioassay confirming that we had resistance to pyrethroids. Here we tested permethrin with a high level of resistance. We also had a little bit of resistance to coumaphos and amitrase. So showing the potential of those bioassays, of this, not the bioassays, but the molecular assays in detecting resistance very fast and with precision.
Dr.
Saying that,
Guilherme,
well, we can wait.
Do you have only 5 minutes for the end of the presentation?
Yeah, okay. All right, no problem. Yeah. So showing the potential of those methods to detect resistance and how they are very powerful. So, and what is amazing is that we can use these molecular markers to develop resistance detection methods in other species of ticks. So for pyrethroid resistance, we know that we have mutations concentrated on the domain 2 of the sodium channel. They are overspread across the different species of ticks in different locations in the earth. So knowing that, we took 8 different species that had access to ticks, and we sequenced. We used those live ticks to extract the RNA and cDNA. We did sequencing using primers based on Exodes scapularis in microplus in the Varroa destructor. So we did a touchdown PCR to sequence those fragments, and we were able to characterize those 2 fragments from different species of ticks, important ticks like the tropical horse tick, Anlatus tick, Amblioma americanum, Amblioma mixtum, sanguineus, and Hyaloma anatolicum. So we were able to sequence that, and with that we were able to describe a mutation associated with pyrethroid resistance in Hepcephalus sanguineus. We were able to describe a mutation associated with pyrethroid resistance in Hepcephalus annulatus. And right now we are working with some populations of Amblioma mixtum from Mexico that show resistance to pyrethroids as well. And we are detecting, using the same technique, the HRM, showing that we have some resistant traits here. We were not able to sequence those genes yet because of the pandemics and all that, but we're working towards finding a marker for resistance in amblyoma, right? So in terms of perspectives, looking at the mechanism of resistance, like to study the detoxification process, we can use transcriptome of Staphylococcus microplus exposed to acaricides. We have a project with Northern Arizona University working with that, and we're developing an NSeq platform for genotyping and a LAMP-PCR for detection of acaricide resistance mutations to spread more and try to enhance the capability of detection, because we know of the potential of these methods for resistance detection. And we also are giving another jump for another species, so we're going to start working with detection of mutations associated with insecticide resistance in Cochleomyia modivorax, which is the screwworm fly to the pesticide. So we're working, this is a work in progress. It's a student from our master program. So with that, towards the end, I'd like to acknowledge and thank a lot of people participating on these results. The team from IPVDF, from the USDA, collaborators in different universities, Egypt, Mexico, Texas, and also our lab students. A lot of this work were made by undergrads and master students that worked with me on the last 4 years. I would like to, of course, acknowledge the financial support of these different agencies. And thank you very much. I'm glad to get any questions. I'm sorry if I passed the time. I guess I had until 7:20, right? No.
Yes, you have more 5 minutes, but for questions. But okay, no problems. The last presentation, I have time. And
it's now, Friday, right?
Yes, it's Friday. I will be initiated with the comments. So Thais Ribeiro, thanks a lot. Dr. Guilherme, for your excellent presentation. And Fabio Scott, thanks to you for participation in the International Symposium. And now you have 2 questions.
Good.
And Dr. Yara Cid make 2 questions. She said, in addition to food and agriculture organizing guidelines, which describe the methodology for the bioassays, are the guidelines that describe parameters for validation of bioassays methods and describe and parameters for precision, accuracy, linearity, and robustness. This kind of describe these kind of parameters.
If the guidelines describe that, that's the question.
Yes.
Well, those guidelines they were produced in 2004. They are more like a material and methods for running those assays. So they give you which kind of paper you have to use, which concentration you need to use for each pesticide. You have to use larvae of 14 days old and all that. So it's more like a material methods part of the paper. You know what I mean? It doesn't give you analysis, too much detail on the analysis, doesn't give you too much detail on the robustness, and doesn't compare bioassays with each other as well. So we have some papers describing like why adult bioassays are less
limit of quantity.
Precise, precise to detect resistance and all that. So the guidelines are more like this is what you got to do to detect resistance. These are the materials, these are the concentrations, and this is the way you do the analysis and do the graphs and do that. And they do a little discussion on the methods to overcome resistance or to manage resistance. That's the basic guidelines for FAO.
And the second question is, do you believe that molecular biology techniques will replace current techniques in vitro?
No, I don't think so. I don't think, because one doesn't, as I showed on that case study in Puerto Rico, the confirmation of resistance is, you only can have a confirmation of resistance when you have the functional assay, right? It's the molecular method. It's a good screening process. It's a rapid process. So if you go to the field, of course, across time, when it comes with time, you see, well, there's a big correlation between efficacy failure and the presence of that specific mutation of that specific genotype or a certain population. But until then, it takes time. And but even though the confirmation of resistance will come with bioassays. So it's a good method for screening. Say, if you go to the field, say, oh, well, I have 10 ticks here collected, like nymphs from cows. And our idea is to have a handheld thing to run the genotyping, right? And you can say, oh, those 10 ticks, they have the mutation associated with resistance to amitraz. I'm not going to use amitraz. But you can collect ticks and confirm that later. But in terms of practical point of view, it's important to have the molecular assay. That's my thought. I don't work to develop molecular assays to get rid of the bioassays. I think they need to work together.
Thank you. And return it to the validated method to the guidelines. And you know, if you have another guideline that have the validated, the linearity, the methodology to make linearity, the methodology to make the quantification limit, detection limits, do you know if you have described this in another guideline?
Not guidelines. So this paper that I mentioned here, that came last week, it gives you a good view. Of course, you have to do adaptation because they are using flies to on that paper, but it gives a good broad view on how to
confirm, to make a test that you have a statistical.
Yeah, how to make a test. Yes, how to choose the groups, how many groups we're going to use.
Yes.
What concentrations are going to use. So there's way to do that. Yes, and there are some the Robertson Priesler. There's a book from the people that wrote the program, the Polo Plus. They have a lot of the specifics there. So there's a book about it. It's called Pesticide Resistance Bioassays in Arthropods. That's one of the books. It gives you like how many dose groups you're going to use and all that. So this paper that came out last week, it's a very cool paper. It's very practical. It gives you like real examples of, I have this flies, I'm going to test these products, I'm going to test. So the range of concentration I'm going to use is from this to this, and then you retest and you do and you cut that line in more points. So you're going to refine the dose response. But there's this paper that came out last week that I put in on the presentation. When maybe we can share here I can share again the screen maybe, and
I think so.
So people can take notes. Go. This one, oral and topical. It came out in the last issue of Journal of Insect Science. So it's a very handy paper. It's very specific on how many dose groups. They give you the on the supplementary material. They give you the codes for R, so we can program R and calculate the LC50s and do the curves and graphs and all that stuff.
Thank you very much. I will consult it.
Good.
Thank you. No more questions. Thank you very much for your participation.